Today I would like to present mostly the work of my past PhD students, Bastien Le Gars Santoni, who has been working with me on a Swiss National Science Foundation project that I submitted with my colleague Paul Bowen at EPFL in Lausanne. And you heard the title before, so I will directly go into the topic. I will start by an introduction on ion-doped calcium phosphate. I would like then to spend some time on trying to explain why it's kind of complicated to produce pure beta-tricalcium phosphate powders and dense cylinders. I will present some osteoclastic resorption results on these dense pure beta-TCP and also doped beta-TCP, and I will finish with concluding remarks. The field of ion-doped calcium phosphate started, I would say, 20 years ago with the work of Bill Bonfield, and probably with this key publication from Ian Gibson, Serena Best, and Bill Bonfield on silicon-substituted hydroxyapatite. Between the year 2000 and 2010, this group published probably close to 100 papers on the topic. And also Bill Bonfield started his own company, Apatech, and was sold for $330 million, which has raised the interest of many other researchers trying to do good science and also to become rich. So there's been a huge interest in this doping, not only with silicon, but also with strontium, with magnesium, with sodium, and so on. And in 2020, I published a review article on beta-TCP, where I indicated in these two tables on the top the reason why you would dope, in that case, a beta-tricalcium phosphate. Some are for biological reason, but also for other reasons like luminescence, and the number of ions that have been taken for ion doping. This gives an idea about the interest from the research community for these approaches. If we come back to the work of Bill Bonfield, one may wonder how the silicon can really, or the silicate can really affect, have a biological reaction or effect on hydroxyapatite, because hydroxyapatite, the sintered version, is pretty inert once implanted in the body. And this is an example of a clinical study by Lienert in Germany, showing basically that nothing happened to the sintered hydroxyapatite after 7 years. So Bill Bonfield always mentioned that silicon or silicates were included in the structure of hydroxyapatite, but if the hydroxyapatite never dissolved, how would the silicon or silicate affect the biological reaction? One potential hypothesis is that the silicates are not present only in the lattice of the hydroxyapatite, but also at the grain boundary. And this group from Paris and Japan showed that actually there are some silica gel units that are present in this silicon-substituted hydroxyapatite. And so the silicon or the silica might be released by dissolution of the grain boundary. And in fact, the grain boundary composition is a very big topic in ceramic. It has not been really addressed in the field of bioceramics. And so we've been observing funny things over the years in my lab. For example, here, two beta-tricalcium phosphate ceramic that are dissolved in aqueous solution. In one case on the left, you see that the grains were more unstable than the grain boundary. So you have a grain dissolution. And on the right-hand side, this is the typical behavior where the grain boundary are dissolved and the grains remain where they are. And so actually we could postulate that maybe on the left-hand side, we would mostly have an osteoclastic resorption because the cells need to go to the surface in order to degrade the material. And on the right-hand side, we might have the release of particles, and then the resorption would be maybe induced by macrophages. So adding contaminants into the structure might have different effect. First, by being released and having a direct biological effect, but also by modifying the way the ceramic is resolved. So these are the topics we wanted to address in this SNSF project. And to start from the start, we wanted to have a pure beta-TCP material that we could use as a control group, and then to dope this material and look at grain boundary composition and solubility, and also at behavior of macrophages and osteoclasts. We use a traditional method for this production. We started with calcium deficient hydroxyapatite that we sintered or calcined at 850 degrees. And we wanted to make sure that we have no chemical contaminants and that we have no secondary phases like hydroxyapatite and beta-calcium pyrophosphate. If I show here on the right-hand side the phase diagram of calcium phosphates, you can see that if you have too little calcium oxide or a bit too much phosphopentoxide in this area, you have the pyrophosphate as impurity. And if you have a bit of excess of calcium oxide, you form hydroxyapatite. So the synthesis was fairly standard. I hope you see my video. So basically, we added the ammonium phosphate solution into a calcium nitrate solution. And at the back, we control the pH with ammonia solution. So in that case, we kept a pH of 8. You can see the fluffy or this whitish powder. This is amorphous calcium phosphate. If you leave it for 2 days at 30 degrees, you will form calcium deficient hydroxyapatite that you can then filter, dry, and convert to beta-TCP at 850 degrees. And so how can you control the phase composition? Simply by changing the pH. Bastien did a lot of experiments and varied the pH from 6 to 9. And if you have a low pH, you form the pyrophosphate, so the beta-calcium pyrophosphate. If you have a high pH, you form hydroxyapatite. And the pH of 8 seems to be ideal in terms of purity. You can also see that there's a lot of variation in the result. So it's not like if you do the experiment once, you will automatically get 100% purity. You need to repeat a few times, but basically pH 8 is ideal. And then the complicated part for us was to achieve a high purity in terms of contaminants. And the first test we did was a 99% pure calcium nitrate and a 99% pure phosphate, ammonium phosphate. And this is the result. The first one, we had 300 ppm of strontium. And Paul Bowen at EPFL always told me that any ceramic that has more than 100 ppm of something is a doped ceramic. So in that case, we would have a strontium doped ceramic. So it took some time for Bastien to find raw materials that were pure enough to produce a beta-TCP that has a very low amount of contaminant, in that case 20 ppm of strontium. And we had no other contaminant like that are listed here. And actually we did a screening by ICP-MS, and we had over the 40 different elements, or 47 I believe. Strontium was the highest amount. So we can say that we have what we could call a pure beta-TCP. And so he did an intensive Rietveld refinement analysis of these powders that he produced. You have here the calcium to phosphate ratio, and here the lattice parameters of the beta-TCP. In this area, you have a bit of calcium pyrophosphate impurities. In the high range of calcium to phosphate ratio, you have a hydroxyapatite. And actually, we would expect that the lattice parameters are constant for beta-TCP because there's only one structure. And we saw this shift of lattice parameters and a jump at 1.5. And so we thought maybe it's a solid solution that happens. We looked at the calcium to phosphate ratio by ICP-MS versus X-ray diffraction. And if we had a solid solution, we would expect a jump somewhere, but we see no jump. It's perfectly linear. Beta-TCP is a very special structure because it has 6 calcium 4 sites that are represented here in green. And these are half empty or half full. So you have 3 calcium that are distributed in these 6 sites. And one of the assumptions is that this distribution is random. but there are also some atomistic simulation that suggests that maybe they are, the filling is according to planes. And the conclusion from looking at this and this structure and some atomistic simulation that I'm not going to show today suggests that actually we have different beta-TCP within the beta-TCP, and they just vary in the filling patterns of these calcium 4 sites. So there are some large domain in the order of 100 nanometer that have one filling pattern, and other domains that have a different filling pattern. If you want to know more, you can check this paper from the JEX early this year. Then the next step was to produce a dense beta-TCP sample. I had the help from the Jean-Christophe Ornay from Université Valenciennes for that. He showed us how to produce the dense sample by slip casting. We sintered the sample. And then we characterize them in terms of grain size and relative density. Here I'm showing 6 samples that we use further on in the osteoclast study. And they vary well, we named them calcium P1 to 6, and they vary in terms of calcium to phosphate ratio from 1.495 and up to 1.502. Here you have about 1% pyrophosphate, and here about 1% hydroxyapatite. So all these beta-TCP powders are over 99% pure, so a highly pure sample. And you can see that there's a clear shift of the grain size and also the relative density at this level of 1.5. If you look at SCM, you see the shift of grain size between below and above 1.5. If you look at the densification, it's almost perfect with a value over 1.5, and then it decreases the more pyrophosphate you have. So it's not that easy to produce samples that have the same grain size, the same density, and that could be considered as identical more or less for the cell culture studies. Then another problem we have is that calcium phosphates evaporates during sintering. They release phosphor species, and if you start with pyrophosphate, you form a bit of beta-TCP on the surface. And if you start with beta-TCP, you form calcium oxide on the surface. And so sometimes these layers can be quite large. For example, with the pyrophosphate, you get up to 200 micrometer in thickness, where you have a different composition on the surface than in the bulk. That means that if we want to really understand what if we want to do cell culture studies with a clear and controlled composition, we need to polish the surface. So Bastien spent a lot of time trying to find conditions to produce enough of those samples that would be polished for cell culture studies, because typically we need a few hundred samples for these studies. Then we started playing also with the sintering atmosphere, which is something that hardly anyone does in the field of bioceramics. And we realized actually we can modify the properties by changing the atmosphere. The result I showed before are these triangles, and now these squares are samples that were started with the same powder but sintered in a phosphor-rich atmosphere. So what we did was to co-sinter calcium hydrogen phosphate and beta-tricalcium phosphate. And the calcium hydrogen phosphate releases a lot of phosphor species and basically saturate the atmosphere. So we had this prevented the beta-TCP to lose phosphor species. And we see that this has an effect on the grain size, on the relative density. And on the right-hand side, I show the composition of the polished sample by XPS. These are the samples that were sintered in a normal atmosphere, so polished. Ratio is slightly below 1.5, but this is normal considering that it's XPS. With this other sintering with monetite, you have a slightly lower value. We still don't really know, quite understand why. And if we take this sample and we calcine them at 500 degrees, we have a bit of evaporation and formation of calcium oxide, and this leads to this small increase of calcium to phosphate ratio. And I would like to move to the osteoclastic results. And And so the idea was to take a dense, pure beta-TCP, to polish them, to have a control grain size, and to use that as a control, where we would then look at the effect of sintering atmosphere, calcium to phosphate ratio, and also dopant. We did some tests with magnesium, with copper, with strontium, and I will only show results with copper because strontium had no effect and we had too few data with magnesium. The production of the osteoclast is given here. We started from bone marrow cells. This is work done at University of Bern. The non-adherent MCSF dependent osteoclast progenitor cells were then cultured with MCSF and Rankel, and then the osteoclasts were suspended and seeded on the material. We had a trap staining to identify that they were trap positive cells. We had an ICP-MS analysis of the cell culture medium to look at calcium and phosphate level, and strontium and copper if we had doping. And then we looked at the resorption, or cleaned the surface and looked at the resorption. This is just an image of the trap positive cells present on the surface, where you see that it's clearly trap positive and multinucleated. The surface of the sample looked like this before the cell culture test. You see some of the polishing line. If you place it in the cell culture medium, you see a tiny little bit of dissolution. Mostly we attribute this to the formation of an amorphous phase on the surface, especially in the polishing line. The osteoclast resorption is activated by a bit of adding a bit of HCl that lowers the pH to about 7.1, and we can see that this affects a little bit the surface, dissolve it a tiny little bit. If you add macrophages, nothing really happened, and if you add the osteoclast, you start seeing resorptive activity, where this needle-like structure and the formation of pits. The measurements of the resorption was done by profilometry. You see here on the image of the surface that you have an osteoclast that moved on the surface here, the same here or here. And we measure basically the volume of this material that was removed, and we normalize it to the surface of the material. So we obtained a mean resorption thickness. With no cells, macrophage, nothing really happens. And it's when you add the osteoclast that you see resorption happening. There are 6 different CAP or histogram bar. It's because we use these 6 different materials. And just to remind you, let's quickly go back here. So this corresponds to this 6 tricalcium phosphate material. And if we look in more detail at the data, we did a statistical analysis. There's a strong effect of the calcium to phosphate ratio below 1.5, which was actually, when thinking about it, not so surprising. So we had an inhibition of the resorption by having a bit more calcium pyrophosphate, which is a strong inhibitor of osteoclast. What was more surprising was the fact that we had a 30% drop with not even 1% beta-calcium pyrophosphate. So you could imagine that with a 3 or 3.5%, you would completely inhibit osteoclast resorption of beta-TCP. And the standard ISO standard says that you can call your beta-TCP pure beta-TCP if you have up to 5% impurity, like pyrophosphate. So this is very important to really, when comparing calcium phosphate, to control this calcium pyrophosphate. We also realized that the sintering atmosphere affects the result. I'm not going to go too much into the detail, but if we use the standard points are the blue triangle, but when we did the sintering and in the phosphate rich atmosphere, we had a higher resorption rate, and if we did it after calcining the surface at 500 degrees to form calcium oxide, we had a drop. These are results with the copper doping. You can see the nice change of color when you add a bit more copper. And if you add too much, well, If you add more, then you get this black color, which is typical for copper oxide. And actually you can see that it has segregated at the grain boundary. And you can also confirm the presence by EDX or by X-ray diffraction. So these samples were placed in the osteoclast study. This is the standard image that we normally see. The blue would be the holes that are created by the osteoclast. With a bit of copper, you see a completely different behavior with kind of wells or big holes that are created, but in most of the surface, nothing else. And then if you add more copper, then you basically kill the osteoclast and there's no resorption anymore. If you look at the copper concentration in cell culture medium, at low concentration, you hardly have anything. With high concentration, you start seeing something. So for example, here, even though there was no resorption, so we assume that the copper comes from the dissolution of copper oxide. The interesting thing is with a bit of copper, we had an increase of the highly significant increase of the resorption amount. And basically the pattern was completely different. We can say here that the copper can have two different effects. One effect is when the copper is removed from the surface and goes directly into the osteoclast from the bottom, or from the cell culture medium, and not directly from the material. I would like to have a few concluding remarks. To say that we had a collaboration with Martha Gallo and the group of Jérôme Chevalier and Solène Tadier in Lyon. They were interested in trying to identify the direction of these needles that we find by osteoclastic resorption, and they found that they are aligned according to the C-axis of beta-TCP, and they also had a look at the effect of magnesium on this and found some interesting changes due to the presence of magnesium. This is the paper that I'm talking about. Then one issue we may have is that if we have, I mentioned we have evaporation of phosphorus species, and if you look at the scaffold like this, you can maybe polish the outside, but there's no way you can polish the inside. And since we form all the time this calcium oxide, we generate alkaline conditions inside the scaffold. And actually Yassine Mazouz showed in a similar system that we can have a fairly alkaline pH occurring in the inside of a scaffold compared to the outside. And then, is it relevant in vivo? We have some ideas that it is, because we provided alpha and beta TCP granules to Sharam Ganati, who did then an ectopic and autotopic implantation and look at the biocompatibility reactions, early reaction to these granules. The granules were either crushed, cleaned and delivered, or crushed, clean and calcined at 500 degrees for 24 hours to produce this calcium oxide layer. And there was a significant effect on this of this calcination treatment on the biocompatibility. We had the idea in the project to try to look at grain boundary composition by scanning TEM at EPFL, because that was the task that they were meant to do. And unfortunately, the scanning TM was not able to, did not have the high enough resolution and to detect changes at grain boundaries. So I talked to a friend of mine, Baptiste Go, who is in Düsseldorf at Max Planck Institute, and he gave it a try, or his postdoc Thorsten Schwarz, and we got really lucky the first time he tested the sample, because the sample didn't explode, which all the others basically did. And then second is he found a grain boundary that you can see here. You see the size, there's 120 nanometer. And going through this grain boundary, you can see that the calcium to phosphate ratio varies. So the grain boundary does not have the same composition as the grain. And also in this sample that was not fully pure, we saw that there was an enrichment in strontium and sodium at the grain boundary. And I would like then to conclude saying that basically producing pure and doped calcium phosphate is not as easy as it may appear. The bulk, the grain boundary and the surfaces can be contaminated. Beta-TCP is a highly used materials, but apparently the crystallographic structure is not as defined as one may think, and possibly beta-TCP is actually a mixture of different compounds. And the sintering atmosphere, the calcium to phosphate molar ratio, in particular pyrophosphate and copper doping, affect the resorption. And I think I will just mention that we had lots of collaboration. Anne will be happy to see Université de Valenciennes here. We had, well, I didn't present all the data. We have also a collaboration with Florence Babonneau. Baptiste Go, who has also an appointment at Imperial College. There's actually a PhD starting in the field with Eduardo Seitz. And then people at University of Bern for the osteoclast, people at INSA, and lots of people at RMS. Thank you.